Dear students, welcome to my channel. The topic is Southern Blotting. Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment. The ability of DNA strand to hybridize from its complementary nature clearly depicted in Watson-Crick's DNA structure model. In the beginning, experiments were carried out to study the renaturing ability of DNA and formation of DNA RNA hybrids for understanding tissue or cell specific expression. Thereafter, hybridization came into picture with the development of props which can detect specific sequences within the target and immobilization of the target sequences onto a solid support like nitrocellulose powder, which further gave rise to nitrocellulose membranes. A demonstration by Southern the DNA fragment separated by gel electrophoresis can be transferred onto a membrane, allowed characterization of DNA by hybridization pattern and by their molecular sizes. Later, this method was applied for spatial identification of targets and blotting of bacterial colonies. Dr. Edwin Southern developed Southern blotting to identify specific DNA sequences. It is a detection technique used to find the target DNA sequences in the DNA sample in the field of molecular biology. The process starts from electrophoresis of DNA molecules which are hybridized in a blotting membrane followed by a transfer step where DNA from gel is transferred onto the blotting membrane. Next we can see the principle of Southern blotting. Restriction endonucleases, which is an enzyme used to break the DNA into small fragments. These fragments are then separated using electrophoresis. The fragments achieved is then classified according to their size, in kilodaltons. Thus, DNA fragments are transferred to the blotting paper where it is incubated with props. Props used in Southern blotting can be highly selective. They can selectively bind with a resolution of 1 in a million and the characteristic to bind to the intended target fragments. The DNA fragments are separated on the basis of size and charge during electrophoresis. Separated DNA fragments after transferring on nylon membrane, the desired DNA is detected using specific DNA probe that is complementary to the desired DNA. A hybridization probe is a short, that is 100 to 500 base pairs single stranded DNA. The probes are labeled with a marker so that they can be detected after hybridization. Next we can see the procedure. First step, restriction digest by restriction enzyme and amplification by PCR. Second is gel electrophoresis, that is SDS gel electrophoresis. Third step is denaturation, treating with the HCl and NaOH. Fourth step is blotting. Fifth step, baking and blocking with casein in BSA. Sixth step is hybridization using labeled probes. And the last one is visualization by autoradiogram. Next we can see the procedure in detail. In the procedure, the first step is restriction digest. The DNA is fragmented by using suitable restriction enzyme. Restriction enzyme cut the DNA at specific site generating fragments. The number of fragments of DNA obtained by restriction digest is amplified by PCR. Step 2. Gel electrophoresis. The desired DNA fragments is separated by gel electrophoresis. Third step is denaturation. The SDS gel after electrophoresis is then soaked in alkali or acid to denature the double-stranded DNA fragments. DNA strands get separated. And the fourth step is blotting. The separated strand of DNA is then transferred to positively charged membrane, nylon membrane, that is nitrocellulose paper, by the process of blotting. Next step is baking and blocking. After the DNA of interest bound on the membrane, it is baked on autoclave to fix in the membrane. The membrane is then treated with casein or bovine serum albumin, which saturate all the binding site of membrane. Sixth step is hybridization with labeled props. The DNA bound to membrane is then treated with labeled prop. The labeled prop contains the complementary sequence to the gene of interest. The prop bind with complementary DNA on the membrane since all other non-specific binding site on the membrane has been blocked by BSA or casein. The next step is visualization by autoradiogram. The membrane-bound DNA labeled with prop can be visualized under autoradiogram, which gave pattern of bands. Here this is the overall steps of Southern blotting. First, genomic DNA is subjected to restriction endonuclease, so fragments of DNA forms subjected to agarose cell electrophoresis. See, high molecular weight can be seen on the top and low molecular weight at the bottom. Then that gel is put in blotting buffer, that is agarose cell. On nitrocellulose nylon filter, blotting occurs. Then probe is added, exposed to X-ray film, and the bands can be visualized. Next we can see the application of Southern blotting. Southern blotting is used in a number of applications. The primary usage of Southern blotting is to identify a specific DNA in a DNA sample. It is mostly used in the identification of viral infection and certain bacterial infections. In recombinant DNA technology, Southern blotting technique is used to isolate a particular DNA. It is also useful in the study of mutation and gene rearrangement. This property is used to diagnose neonatal disease and genetic disease. Due to the precision in DNA identification, this technique is used in phylogenetic studies, paternity and maternity analysis, forensic studies and personal identification. Southern blotting is a technique used to detect DNA given sample. DNA fingerprinting is an example of Southern blotting used for paternity testing, criminal identification, victim identification to isolate and identify desired gene of interest. Used in restriction fragment length polymorphism to identify mutation or gene rearrangement in the sequence of DNA. Used in diagnosis of disease caused by genetic defects. Used to identify infectious agents. Dear students, thank you for watching. See you next class with another topic. Don't forget to leave a like and please subscribe my channel.